Hi, everybody. It's a great pleasure to meet you all. As the chair of the session alluded to, Cassandra unfortunately couldn't be here today. She had a family emergency, so she flew back. But I promised she was here for 30 hours in Cagliari. But again, she had to go back to the States. That being said, again, it's my honor to introduce you this work that we recently started in collaboration again with Cassandra, and talks about charting fetal ANS development from at-home monitoring. And to get us started, we decided to put this kind of overwhelming slide to open up, but the idea is to make a point about the fact, and this has been alluded by many speakers from yesterday and today, about the fact that the system we're looking at, it's really complex, and it deals not just about, with the fetus itself, but also the mother, the placenta, and more so, it's just not about the pregnancy, but overall, it is about, again, the pregnancy itself, early life, but also further neurodevelopmental outcomes. And the idea here is the fact that what Cassandra and I have been trying to get a sense of is trying to understand the so-called brain-heart axis during pregnancy, and how this evolved throughout pregnancy, with the idea that by charting this development, we cannot just learn about pregnancy itself, which of course is important and speak to birth outcomes per se, but also the fact that we may, or this may offer the opportunity for investigating long-term health, physical and mental health outcome later on in life. And so in order to do so, what we've been thinking about utilizing is an approach which is slightly different than what the clinical approach is by itself, but rather start thinking about going outside the clinic and learn about the fetal development at home using fetal ECG. And here we decided to put this quote from a paper which is now 15 years old, and it talks about the limitation that at the time people envisioned in this review, which is quite cited, about fetal ECG and its implementation. And at the end of the day, some of these same challenges remain today, and I'm just going to summarize at least a few here, which is pings or at least underline some of the same concepts that we've been discussing. So for example, the lack of publicly available data set for training algorithms, but also the fact that we have a wealth of devices that we can utilize both in the clinic and at home. We don't even necessarily know yet how this different position and combination of electrode may play a role in detecting fetal ECG, as well as the fact that, and this is something everybody keeps talking about, how data integration from different modalities, not necessarily just monitoring the electrical activity, may play a role in getting a better understanding of the fetal heart functioning, and how, again, that we can extract meaningful data from that signal. Building on this, we do also have limitation in trying to understand this from a research point of view, not necessarily sticking to the more clinical approach, which is the fact that antenatal monitoring is usually conducted in clinical settings. We heard great talks today about how to bring that outside the clinic, but I think we're definitely not there yet, so that's what we've been trying to spearhead a little bit. And also the fact that in the scenario where we are right now, definitely we do have a sparsity in data collection. There's definitely some gestational age week which is over samples versus others which are a little bit less investigated. In order to try to address some of this, we have started thinking about a slightly different study design and aims to be able to answer some of these questions. And so the idea here, without getting too much into the weeds of the study design, but again trying to get a better sense of how we wanted to do this, is looking at the evolution of this trajectory of fetal ANS development in a death sample period of time during gestation. So starting from 20 weeks gestation up to 34 weeks, doing that once a week, so really then sampling and evenly distributed in this range, with the idea of testing this first aim, which is how can we actually do this reliably, how can we actually collect fetal ECG in the home environment, and how we can extract meaningful measures out of that. And without going too much into the details of the broader aim of the study, but this talks a little bit of what I started the presentation with, it's really trying to understand the role of some of exposure and stressor that may change or get some of this trajectories of development a little bit different from the, let's say, normative trajectory. And in this sense, one very important exposure that Cass is particularly interested in is maternal stress and how that can impact fetal development and whether different trajectory of stress itself may play a role. And ultimately, pinging back or alluding to the overall aim, which is learning more about the heart-brain axis, and this is something more in the expertise that Cassie brings to this project is the fact that at the end, so towards 34 or 35 weeks, depending on also when participants are available, there's also a fetal MRI done, so really trying to get this sense of like brain heart axis. In order to do so, we have been testing 2 different devices for monitoring fetal ECG at home. I want to put a disclaimer here that doesn't necessarily mean there are other options. We've heard today there are other great options. Here is where, of course, science meets a little bit logistical aspects and availability of devices, and so these are the ones, the 2 we have tested. So this is called, well, the one on the left-hand side of the slide is called Nuvo Invo, and it consists of 2 belts, which are sitting one on the top and one at the bottom of the birthing parent's belly. And talking about data fusion, which is what I was alluding earlier, this device does not only collect the electrical activity of the heart, but also has acoustic sensors, which the way they advertise it or the way they explain this is the fact that they're able to complement the missing information coming from the electrical activity, or when, I mean, I think more precisely, when the electrical activity is not so clearly detected, then the acoustic one can basically fill in the gaps. And then the second device is this one on the right-hand side of the slide, which is a more traditional fetal ECG recording. 4 channels, it's on, the belly has right here, and is only looking at the electrical activity. One disclaimer, which I think it could be a good point for discussion, or in general something that I also, as a presentation, alluded to, since we're working with clinical/research devices, doesn't necessarily mean that there's always an opportunity for being able to get a sense of the algorithm. And so while we're not dealing necessarily with black box approaches for detecting fetal ECG, it's definitely true that we're also not dealing with open source approaches. And so that, of course, comes with limitation, especially if group doesn't necessarily have expertise for being able to extract the signal itself. I think it's worth mentioning that we have, in a way, trust a little bit what these companies are providing us, and I think, again, it's important more for a point of discussion for later. In terms of the data we'll be presenting in the next few slides, again, the idea was participants, or the initial participants that we have in this study were at the beginning stage, so we will not have a huge sample size, but definitely we're looking for input and feedback as to how we're doing so far. Is that we're asking participants to do this recording once a week. We're aiming for 30 minutes of fetal ECG recording, and in the next few slides I will show you some preliminary results related to data quality and some metrics which have been extracted from this. So in terms of pre-processing, pretty standard, so we're getting the RR series, we're computing some parameters here. Of course, it can be more onto this, but what we're going to show is some time domain parameters, which we'll see in the next few slides. Before getting into the actual measures and again feature that could be extracted, here is some sort of like summary of data collection or compliance, which is something also we're alluded earlier today, and I think it's important to keep in mind if we were to want to move this forward and try to implement it outside the clinical setting, how participants are reacting to this. So in this slide you do have here the device, which in the previous slide was on the left hand side, bottom it was the right hand side, and we're talking about number of recordings here. So you can see this 87% and 67% as a very rough, but I think insightful measures of how this compliance was achieved. And for successful recording here, we allude to the fact that, or at least we consider a successful recording if at least 5 minutes worth of data or good data was collected. And again, not to make too much out of this because we're dealing with 15 and 15 recordings, not too many, but we're getting slightly more data for the one device, again, the Invo one. And just a disclaimer, we'll see more in the next slides, with these 15 and 15 are not individual participants, because again, as we saw, like, we're aiming for longitudinal, so one given participant may have contributed more than one recording. This is a slightly different way for visualizing data usability, and so as we can see here, here in this graph, we do see a nice concentration of usable data within 20 and 30 minutes, which is what we're aiming for. We do have, of course, the left-hand side of the distribution when participants could only contribute a little bit less data, and also you have the usual overachiever participants, which went above and beyond, so collected even more data than what we asked them to do. There was no correlation, or at least at this stage, with the data quality and some of the measures that are going to be described, which I feel is reassuring, again, to be determined when we add more data. Here is a graphical depiction of, again, what different time points that different participants have contributed. This is the 5 participants we have. You can see the 2 at the bottom. This is a little large, but I hope I'm able to direct your attention there. I've contributed data at many different weeks, so each one, these little dots, displays when the fetal ECG collection was attempted. You can see those in red, meaning, again, they were usable section, and those in gray were attempted sessions, but the data quality was insufficient. And going back to some of the discussion earlier, we also have noticed definitely a better compliance in terms of collection of data when we're able to give participants some sort of feedback about their sessions. And so here is an example, and I apologize, a little tiny, some information about their session with just one number talking about the average fetal heart rate during that session, fetal maternal heart rate during that session, and then some more, I would say, less sciencey but still informative for participants. Let's say, well, what the size of the fetus compared to some object or fruit or stuff like that, and really keep them more engaged. All right, getting to the data. So here I'm showing 3 different features extracted from this recording. So from left to right, you do RMSSD, SDNN, and fetal heart rate. Each of this point is contributed by 1 session, so that's why you should expect in the same 23 points as previously displayed. And again, it's a small sample size, but it was so, or at least we're starting to see some of reassuring trends that we know from some previous studies about the developmental evolution of some of these measures, which again speaks to the fact that we are able to get reliable data out of this, with, of course, a grain to be taken with a grain of salt given the sample size. But also we're already starting seeing how in some gestational age weeks where we know that collecting data is tricky, we're seeing that definitely there's worse data quality in there, and it will be interesting to moving forward to see whether or not the devices which is in both electrical and acoustic information may able to fill those gaps a little bit better. Now, one point which was brought up also yesterday is how we can utilize something we know from physiology a priori knowledge to be able to clean this data a little bit and try to better understand variability. It definitely something we are interested in implementing. This is the fact that accounting for sleep states may definitely achieve 2 aims, which is, again, improve the precision of some of the measure by not mixing too many different stages at once, but also the fact that investigating this evolution of sleep stage across the station may implicitly give a more or a better understanding of neurological development on top of just the heart development, which is, again, brain axis is what we're really interested in. And to wrap this up, I think a few more. Okay, 5 more minutes. I also want to leave enough time for a question. Again, these are graphs of the same measures, so fetal heart rate, SDNN, and RMSSD for those participants. Let me go back quickly just to recontextualize this for these 2 participants at the bottom, which are contributed at almost 15 sessions each. And without, again, taking too, or abstracting too much from this, I think it's worth mentioning the fact that participants are contributing different trajectory throughout gestation, and I think a big or a missing piece of this is how to interpret this in the bigger context of learning about trajectory of development. So participants different baseline values, a different, again, trajectory themselves, and the idea is that how we can learn from this and talk more about their development and contextualize this as also using some of this trajectory as predicted for future outcomes. So to wrap this up, want to bring up a few points, again, mainly for discussion. I hope we were able to talk a little bit about this utilization of fetal ECG in the context or outside the clinic, trying to bring this outside of that. We hope to continue to ensure rigor and reproducibility of the data we got so far, but again, reproducibility is also to be taken with a grain of salt in the sense that different participants do different things and want to understand how that can tell us about development. One point which I know many are interested in, we definitely also want to integrate the data we got so far, is the fact that, again, one very important piece of this is learning more about the mother and how that physiology may have an impact on fetal development, and definitely we do have opportunities for doing that with wearable devices and also by collecting additional contextual information to be able to contextualize the estimates that we're getting. And lastly, I think this speaks more to the overall aims of this project, which is investigating sensitive window and really try to, by starting to at this time sampling, understand whether or not there are more parsimonious way for collecting this data and maybe focus energies in some weeks or a subset of the weeks which you feel is really important to have a better and more in-depth phenotyping versus, again, trying to aim for the entire gestation, but I think it's still valuable at this stage since we're in this initial stage investigation. So with that, I want to thank you for your attention. There's many more folks than the one we have here on this slide. And also shout out to the participants who have contributed their time and all their energies to this research study. So it's really, really important to see interest in this project. So thank you all.
Thank you very much, Nicolò.